we're not going to wait for much longer. I mean, I'm not going to give a second introduction, just to say that we had a beautiful conference last week, and that we're very happy to have Jim again today.
Yeah, I'll just put
We will give a conference on the logic of Golgi's apparatus, continuous purification of proteins in the cell. Thank you, Jim, for being back with us. You have your own
I have my own microphone. Thank you, Alain. Good. So there are a number of neuroscience friends in the audience today. I hope you're not too disappointed in the fact that this is a general topic in cell biology, but you can console yourself in the fact that even nerve cells have a Golgi apparatus, number one, and it's named after, and indeed was discovered by, one of the great neuroscientists, historical figure, Golgi. But we've learned a lot more about the Golgi apparatus named after him. And in fact, it was discovered by Golgi. Some of you may know Golgi really pioneered impregnation with metals and metal stains that were important in early neuroanatomy at the light level. And the Golgi body was a mysteriously staining object in the cell body of nerve cells, and it was, of course, in many other cells. Its significance was completely obscure, and it really wasn't until George Palade and Marilyn Farquhar in the late 1940s and early 1950s applied electron microscopy in combination with Golgi silver stains that they were able to identify the ultrastructural equivalent of the Golgi apparatus, and they discovered that it had a very characteristic and unusual structure, consisting of flattened cisternae of membranes. Each of these is a membrane vesicle, flattened, and these flattened cisternae, with each composed of lipid bilayers, are stacked onto each other in a characteristic way, and it's developed that this structure is a universal feature of all eukaryotic cells. The Golgi apparatus is typically located adjacent to the nucleus. It is equipped, we now understand, with microtubule-based motors that constantly power it to the centrosome, so that's why it's at the nucleus. The basic element of the Golgi is the cisterna and the stack of cisternae. Typically there are 5 or 6, sometimes a few more, sometimes a few less, cisternae in the stack. The stack are adhered to each other in their flat centers, and their rims are typically bent. And you see numerous vesicles of the sort that I showed you on Friday that Palade first identified and suggested might be involved in trafficking between the endoplasmic reticulum and the Golgi, but also suggested might be involved in transport in some fashion within the Golgi stack. So historically, what we knew about the Golgi was that before the classic work of Palade and colleagues was that was its ultrastructure, the stack of cisternae. What Palade taught us is that secretory proteins go through the Golgi, and I emphasized that the other day. They go through the Golgi on their way to storage in secretory storage vesicles for the cell exterior. In the 1970s, after Palade's main work, others discovered that as proteins of various other compartments became characterized, proteins of the lysosome, proteins of the plasma membrane, as they were individually cloned and characterized and immunolocalized, what we learned was that the proteins, the precursors to proteins that reside in compartments like the lysosome, vacuole, endosome, apical plasma membrane, basal plasma membrane, and so on, we learned that these are all synthesized in the endoplasmic reticulum. They have global signal peptides. They then pass through the Golgi, and where they can be found in significant amounts, but very transiently on the way to their permanent destination. So we learned that the Golgi is a sorting station. We also learned something very striking initially from the work of Kai Siemens. And I'm now bringing you up to the mid-1980s, early mid-1980s. What we learned was that the proteins targeted to post-Golgi locations, lysosomes, plasma membranes, secretory storage vesicles, they pass as a mixture somehow through the Golgi together. And what's very odd about it, and I'll come to this in more detail, is that even at the very last station of the Golgi, called the trans or mature phase, where the proteins are distributed to final locations, there's still a mixture of proteins. So you remember yesterday, on Friday, I referred to types A, B, C, and D proteins. What I'm telling you is that A, B, C, and D are deposited into the ER, and they transit as a mixture, A plus B plus C plus D, all the way through the Golgi. And it's only when they leave the Golgi, at what Kai Siemens called the trans-Golgi network, that they're separated according to their destination. If so, what was the purpose of the passage through the Golgi? Well, there's another line of discovery that came in the 1960s concerning the Golgi, and it traces to the Canadian histochemist and anatomist C.P. Leblanc. And what Leblanc did is discovered that sugars are added to proteins in the Golgi. We knew, of course, that many proteins passing secreted from the cell are glycoproteins, and we knew the chemistry of many of their linkages, but it was Leblanc who pointed the way to indicate that these sugars are histochemically found on these proteins, on similar proteins in the Golgi, and this is where proteoglycans and related areas, related molecules are found. This led Fleischer and Palade independently to discover that glycosyl transferases, the enzymes that add these sugar linkages, are primarily found within the Golgi. And so we had a new dimension then to the function of the Golgi. One dimension has to do with somehow the maturation of secretory, and now we know many other proteins, to other destinations, A, B, C, and D, in the cell. And many of these proteins are glycoproteins, and these sugar residues are now added within the Golgi. So the Golgi, therefore, has two overall functions that were recognized: glycosylation of sugars and to these proteins, and somehow the passage or maturation of secretory proteins and related proteins. And again, just a bit of anatomy. The cis face, which typically faces the endoplasmic reticulum, the trans face, which oftentimes is away from the endoplasmic reticulum. Vesicles arrive by fusion from the endoplasmic reticulum, joining the cis face. They leave the trans face. And the question that I will largely address today is how they get across the Golgi stack, and in a way, what is the purpose of their moving through the Golgi stack. So what exactly goes on in the Golgi? Now, I became interested in this problem actually as a first-year medical student when I had a histology course, and the Golgi apparatus was presented, and it had this fascinating structure, and nobody really knew anything more than what I just told you about it, actually even less, because it was 1971. But somehow the idea that you have this flattened cisternal geometry in a stack is so it's somehow compelling. And so when one seeks to understand the function of the Golgi, we have to understand in a basic way how its form suits its function. What is it so special? What is so special about a stack? Why is the Golgi apparatus stacked in virtually every organism, whether it be plant, animal, or microbe? So there's something very fundamental about this, and if we can't understand why there needs to be a stack, we really don't understand the Golgi at all. And so to recapitulate then, the key questions are, at least as I began working on this problem in the early 1980s, how can it be, what is the purpose of the Golgi when the very same proteins that need to be separated from each other according to their destination pass as a mixture through the Golgi and are not sorted at all? So why did they need to go through the Golgi at all? Well, one explanation would be that they needed to get modified by their sugar residues, and that could be the explanation, and many people feel that that is. I have never felt that that was the correct explanation, because I still don't understand why those modifications couldn't take place in a single compartment, for example, the endoplasmic reticulum. Why do they have to take place in successive compartments? And as a matter of fact, the drug Berfeldin A, some of you know, causes the Golgi apparatus to fuse with the endoplasmic reticulum, and when that happens, the processing of these same saccharide chains takes place in a perfectly correct way. So apparently they do not need to be partitioned into different compartments, even though they do. And again, the other large issue, what is the purpose of the stack of cisternae? And what I'll describe to you today are the modern developments of an idea which I presented in 1981 as the distillation hypothesis. And the basic idea is actually very simple. The Golgi apparatus is acting like a protein purifying machine. It is acting like a chromatography column whose theoretical chromatographic plates are the plates of the cisternae of the Golgi. For those of you who have seen petroleum refineries, you've seen which act to separate oil into its constituents by continuous fractionation. This is done by a series of mixing and demixing operations in a series of closely opposed plates. For those of you, that's the basic idea. So the basic idea is that the endoplasmic reticulum exports to the Golgi a crude export mixture. That crude export mixture contains a mixture of those proteins that need to be separated from each other for final destinations, and that this occurs, as Kai Siemens found in 1981, during the exit from the trans-Golgi cisterna. And what I proposed was that actually something was going on during all this, during the earlier passage to the Golgi. It in fact also is a continuous process of protein purification in which proteins that escape from the endoplasmic reticulum, either by accident or by design, are systematically brought back layer by layer by layer. So the basic principle is that a crude export, and this is my diagram actually from 1981. As you can imagine, this was not immediately accepted by my colleagues. It actually is probably still, for the most part, not accepted, at least by the name distillation, which got such a bad reputation that I could no longer use it in my publications for 10 or 15 years. It is now called, and well accepted by virtually everybody in the field, iterative sorting. So that is the politically correct term for distillation, but it is in fact distillation. And what the idea is very simple. A vesicle, we now know it to be the COP2 vesicle. It wasn't discovered until another 15 years had gone by. Exits from the ER carrying the intended secretory cargo, meant proteins A, B, C, and D, but in addition, other proteins, the ones that are shown here without a black dot on them, that are ER resident proteins that need to stay behind in the ER, but they unavoidably got imported into the vesicle. And as a result of that, the first cisterna is filled, according to this model, with a large number of endoplasmic reticulum proteins that got into the vesicles that shouldn't have gotten there, so to speak, or perhaps you needed to put them there, but in any case, they need to go back. Otherwise, the ER would lose its character, and the Golgi would take on the character of the ER. I hope I'm being clear. And the other basic idea is that these ER proteins are so prevalent, some of them anyway, that they can't be brought back entirely in a single step. So as a result, what couldn't be brought back in the first cisterna is then brought back in the second cisterna. And what couldn't be brought back in the second cisterna, well, there's a third chance in the third cisterna. So it's a statistical idea. Let's say in the first cisterna, you bring back, you're 90% efficient in bringing back a protein. So now, as this cisterna moves its content to the next cisterna, you have a 10% error rate of ER proteins going forward. That would be absolutely intolerable if it were to reach the last cisterna and be distributed to the plasma membrane. So now you have a second chance, and 90% of the remaining 10% would be brought back, leaving now a 1% contamination rate. And at the next cisterna, I'm making these numbers up because we don't know them, but just to illustrate the idea, it would be 1%. This is called iterative sorting. It is no different than in organic chemistry. You do a differential extraction by partition. You have an oil and a water phase, and you partition them, component A and component B between these two phases. Component A, let's say, goes to the mobile phase, goes to the upper phase, and B to the lower phase. You now have extracted A from B. You have distilled A from B. So you probably in organic chemistry took the upper phase and mixed it with a fresh lower phase and removed some of the impurities. That's exactly what these vesicles are doing. And so without going into every detail, and I think you can understand why the cell biologists in 1980 didn't like this idea, it was sort of physical chemistry, and they didn't like the idea of reducing sort of the Golgi apparatus to sort of a physical chemistry process any more than the bioenergeticists like the idea of reducing high-energy phosphate to a membrane potential. It was just not an idea that was readily understood, I think. In any case, the idea is very simple. These act like the plates in a distillation tower, and the export of the Golgi, which is meant to be a mixture of proteins, is successively purified at each layer by the successive repeated distillation removal in what is called a countercurrent or cross-current a pattern of flow by chemical engineers, is removed systematically. That's the basic idea. And today, just to take a step ahead, this vesicle has been identified. It's the COP2 vesicle. This vesicle has been identified. It is the COP1 vesicle that I spoke about on Friday. The escaped ER proteins have been identified. They fall into two categories. I'll show you momentarily. One escapes by accident and another by design. The one that escapes by design reaches most of the way through the Golgi stack and in fact needs to be retrieved. The system of retrieval has been demonstrated in our work and others to extend right up to the very last cisterna. There are receptors known that mediate this retrograde retrieval. So although none of the protein machinery was known at the time of this proposal, which was made purely on a logical basis, all of the components are known, and the predicted process of retrograde transport was subsequently discovered. What was also artfully avoided here was any discussion of how the proteins move, the secretory proteins, or in general the content of these cisternae will move from cisterna 1 to 2, 2 to 3, 3 to 4, which we call anterograde transport today. And that was not suggested because it was not known at the time. The vesicles that may mediate anterograde transport were not discovered by my laboratory for quite some years. Okay? So that is a sort of slightly modernized view of the distillation hypothesis. So then what are the proteins that are distilled? Now looking backwards from 2015, what are the proteins that are distilled by this molecular machinery that we now understand? The first to be discovered by Hugh Pelham in Cambridge were resident proteins of the lumen of the endoplasmic reticulum. And these proteins, and we now know many of them to be molecular chaperones. So I think we all understand that proteins that are injected into the endoplasmic reticulum need to fold up. And like all proteins in the cell, they rely on molecular chaperones to achieve this. Those molecular chaperones are mostly soluble proteins in the lumen of the endoplasmic reticulum. They need to be retained there. What keeps them out of the vesicles, the COP2 vesicles that leave the endoplasmic reticulum? The answer is nothing. Nothing intrinsically. They're in the water. The water is in the volume of the vesicle, so of course they get out. But nevertheless, when you localize these proteins, they seem to be present in the endoplasmic reticulum. What Pelham discovered was a dynamic cycle in which these proteins are actually vigorously exported from the endoplasmic reticulum into the Golgi, and then actually returned by a receptor from the Golgi to the ER. He called this receptor, which he discovered, the KDEL receptor. And the reason he called it the KDEL receptor is it recognizes the specific sequence, a signal sequence, of 4 amino acids that are present at the carboxyl terminus of the proteins that need to remain in the endoplasmic reticulum. So these soluble endoplasmic reticulum proteins are exported by accident. They are simply dissolved in the water. The water is, of course, transmitted with the vesicle because the vesicle has volume. Okay? If this were endocytosis, we would call this pinocytosis, or bulk fluid endocytosis. Okay? So this is accidental export. The KDL receptor, in fact, binds directly to the COP1 coat protein, and it leads to the effective retrieval of the KDEL containing cargo. Subsequent to this, there were discovered other signals that are present in membrane proteins on the cytoplasmic side, which are exposed on the cytoplasmic side of the endoplasmic reticulum membrane. And in this case, the codomer protein is the direct receptor for those proteins. So, but either way, what we have are components that are needed to function in the endoplasmic reticulum membrane. They have to function there. They are not needed to function anyplace else. You want to retain them there, and they need to be pulled back by retrograde transport. So that's an those are the first examples. Those are the proteins that are removed largely in the first and in the second cisterna. Very little actually gets beyond the first cisterna, which raised the question, what proteins, if any, do you need to have retrograde transport for in later cisternae? And in fact, I have to say, when it was discovered that these proteins, KDEL and similar proteins, are largely retrieved by the time you go through the first or the second cisterna, doubt was cast again on the idea of iterative sorting, because there were no proteins that were known as permanent residents of the endoplasmic reticulum, at least proteins that anybody focused on, that there were no proteins that anybody thought penetrated further and then needed to be retrieved. Okay, they just simply weren't known. So that suggested that there must be something, maybe the hypothesis was incorrect for that reason. Well, it turns out those proteins are now known. And they were discovered in the early 2000s by Lelia Orci and myself and our collaborators. And interestingly enough, they are snare proteins. Okay? And why snare proteins? Well, it's kind of interesting. Here's a vesicle leaving the endoplasmic reticulum. It needs to have V snares, right, in order to fuse. The V snares are highly concentrated. They bind to the coat protein COP2. We in fact know that every COP2 protein binds one copy of the relevant V snare. I won't give you gene names. And Sheckman and John Goldberg have actually solved crystal structures of the V snare bound to this coat protein. There are something like 100 of these copies of the COP2 coat protein per vesicle, and there are therefore about 100 copies of the relevant V snare in every vesicle. These are proteins that need to remain in the endoplasmic reticulum. They have to be brought back in order to allow this vesicle to reform, right? But they are not released from the ER by accident. They are released by intent, because the COP2 vesicle needs to have large numbers, not small numbers by accident, large numbers on the order of 100 per vesicle, one per coat subunit, in order to form the vesicle and to allow the vesicle to fuse. Now we have a certain problem, because the density of this V snare is such that it becomes the major protein in the membrane of this vesicle. And in fact, immuno-EM studies that Lelio and I have published some years ago measure the concentration of these ER-derived snare proteins in the first Golgi cisternae, and in the compartment that forms it, which is called the intermediate compartment or vesicular tubular cluster. And this concentration is fully 50% on the average, half the value as it is in the forming COP2 vesicle. In other words, half of the membrane of the first cisternae of the Golgi is actually escaped endoplasmic reticulum protein, not escaped by accident. So there's no way that you can bring it all back in a single step without bringing back essentially all of the volume. And so as a result, multiple steps of retrieval are necessary for that very reason. And the consequence of that, we think, is our observation that these ER snare proteins penetrate at the 10% level already in the third cisternae, and are measurable in trace levels already in the fourth cisternae. So the conclusion is that retrieval of endoplasmic reticulum proteins probably continues right the way through most of the Golgi stack. And my personal guess is that the reason you have the number of Golgi cisternae that you have is in part to allow the complete retrieval of these vital snare proteins, which are providing the molecular identity of these membranes. Okay? Now there's a third class of proteins that are subject to distillation or iterative sorting, and these are the glycosyl transferases themselves. You remember I mentioned at the beginning that one of the principal purposes of the Golgi is to modify the saccharide linkages of proteins that pass through it. And this occurs in an organized way. In fact, as we discovered in the mid-1980s, the enzymes of the Stewart-Kornfeld pathway that manufactures these saccharides are partitioned into distinct regions of the Golgi along the cis-trans axis. In subsequent work, the most penetrating by Graham Warren and his colleagues, localized a series of these enzymes in the same Golgi in the same cell. Here, for example, is the concentration in the this is the cis-cisterna, trans-cisterna. This is the enzyme that transfers galactose. You see there's a peak. This is the enzyme that transfers N-acetylglucosamine, and not shown here would be the enzyme that transfers sialic acid. These are the proteins are moving in this order, cis to trans, and they encounter these enzymes in the order in which the linkage needs to be added structurally. What I'd like you to notice is that these are not precise distributions. There's a peak that runs across multiple cisternae. These peaks can be shifted by overexpression. So there's actually a dynamic equilibrium of forward and backward transport that is maintaining these distributions. And in a very elegant study by Glick and Oster in the late 1990s, they were able to take reasonable parameters and actually generate, from simple models of bidirectional transport, very similar chromatographic distributions. So the conclusion is, in all likelihood, the Golgi apparatus is engaged in a process of repetitive sorting mediated by COP2 vesicles that enter at the cis face and COP1 vesicles that move at the rims in one or another pattern, some of them back to the endoplasmic reticulum, that can give rise to a process of what amounts to distillation, the subject to which are the cargo, the protein contents of the Golgi. Some are returned to the endoplasmic reticulum effectively, the ER residence. Others are allowed to be transmitted to the transmost cisterna of the Golgi, the mixture of A, B, C, and D that need to go on to other locations, and by mechanisms that I won't discuss today, are then sorted individually according to their final destination. But I would like to focus on today is how the underlying distillation process works within the Golgi, because this point brings us really to the current state of affairs in which there's still a lot of debate about how protein gets across the Golgi. So in other words, there's actually no debate in the field about how proteins go back from the Golgi to the ER when they go back. They go back in COP1 vesicles. But there's quite considerable debate about what the pattern of flow within the Golgi may be. And what are the various possibilities? I will come to that in a moment. This is the first time I've ever given this lecture, depending on how it goes, possibly the last time. And so I'm a little unpracticed about this. I wanted to say one more thing, which is, what is the underlying mechanism of distillation? We know a lot about the molecular machinery, so how can the molecular machinery be used? Well, one possibility is that you have, and you'll see I'll emphasize this possibility, but it's by far not the only possibility, is that you have one category of vesicles moving anterograde and another category of vesicles moving retrograde. And actually, we think that both of these vesicles are coated with the same coat protein, COP1. And the vesicles that go anterograde in this model in green would have to have a systematic way of moving in the anterograde direction, which means cis to trans. The retrograde vesicles, by definition, would have to be moving trans to cis or Golgi to ER, which is not designated here. How could that happen? Well, what Lelio and I discovered, actually, is that there are two separate and distinct snare complexes that are found in every Golgi apparatus from yeast to plant to man. And it's very interesting. There are 4 snares that compose these 2 separate snare complexes. And each is mutually exclusive when it comes to fusion. If you put the V snare of one in a liposome and the T snare of the other in a different liposome, they will ignore each other. If you put the cognate V and T snares in separate liposomes, they will immediately fuse. These are 2 completely functionally separate snare complexes. And yet they are both found throughout the Golgi. And what's interesting about it is that the V, well, I'll call them the cis and the trans snare complexes, because V-cis and T-cis are both found throughout the Golgi, but they're in gradients. And I'm summarizing for you a massive amount of work that I will not show you that goes back quite a few years. It goes back to the early 2000s, 2000 to 2005, where we identified, cloned, sequenced, expressed, tested these functionally, and then immunolocalized them and measured their relative abundance and absolute abundance per Golgi in living cells. So there's a lot of evidence to support this, but as a cartoon, the cis and the trans complex are found in this distribution within the Golgi, and the trans complex, V and T, V trans is found in this distribution, but T trans is found in a uniform distribution. The functional consequence of this is that while T trans is found in every cisterna equally, V trans is found in these gradients, and so fusion will occur primarily in this direction. So if you're a vesicle and you're enriched, for example, in a V or a T snare of the red category, you're going to have a higher probability of fusing in this direction than you will in the other direction. And in general, although it may not be a perfect unidirectional transport, you will have a biased random walk going towards the trans face. Likewise, according to this distribution of snares, if you're a vesicle enriched in the antegrade category or trans category of snares, on the whole, you're going to be moving with higher probability in this direction, chromatographing towards up a gradient towards the source, as it were, of these snares. Okay? So, and this is an implication that comes from morphology. And in a way, this implication points out, and I will come back to this at the end of the talk, fundamental limitations in the field, which are we can't track these vesicles. So everything that I'm going to tell you today is an inference from a genetic test or a morphological test or some, as I'll show you in a moment with unpublished data, or mostly unpublished data, some synthetic biology perturbation of the Golgi, and we see what happens, and then we make an inference from it. Of course, what we would really like to be able to do is track each of these vesicles independently and say, oh, there's one. It has, by the way, so many copies of this snare, so many copies of that snare. It has, by the way, this particular codomer, or it doesn't. It has this cargo, but not that cargo. Oh, and by the way, it left the third cisterna and it traveled over to the second. Well, it did that 60% of the time because the other 40% of the time, vesicles that were otherwise identical went to this cisterna. Oh, and maybe an occasional one fell off the stack and rejoined somehow, or whatever, okay? That is not technically possible because no one has solved the problem of multicolor super resolution light microscopy in 3 dimensions with 100 millisecond or better time frames, which is, in fact, exactly what we're working on in the cell biology department at Yale, and I'll give you some examples in that direction at the end. But that will need to happen before we can actually directly observe these vesicles. As Pilati famously said, or actually it was attributed that Pilati famously said, because I don't know anybody who actually heard him say, that vesicles do not come with arrows, and he was referring to the electron microscope, of course. Okay? So this is, however, a simple way of thinking about it, and what we know of the molecular anatomy is consistent with iterative sorting and distillation working by a kind of chromatographic process or biased random walk, which is, would be underlying this countercurrent flow. Now, in order for this to be effective, obviously these vesicles can't fall off. If you think about it, if this vesicle falls off every time and it can rejoin at random, it's not going to be very good at iterative purification because it lost track of what layer it came from. You can't do that, okay? So these vesicles in the Golgi, in order for distillation to work, unlike every other vesicle in the cell, have to be retained. Now, some of you have probably heard of protein tethers. So a vesicle buds from the ER and it joins the Golgi and it's first captured by a fibrous tether. A vesicle buds from the Golgi, it joins the plasma membrane, and it's first captured by a tether that binds a Rab protein on the surface of the vesicle. Very important general idea. Wrong for the Golgi. Because the Golgi vesicles never actually leave. I mean, they may leave occasionally, and you can measure this, but by and large, they tend to stay. If you incubate the Golgi in vitro, you form the vesicles I talked about Friday, they stay on the Golgi stack unless you extract them with very high salt. Lelio and I discovered why that was quite some years ago, 1998. We observed that every one of the Golgi vesicles is attached by a rod-like filament. This was before tethers were discovered. In fact, we purified what turned out to be the first such filament called P115, which is a component of a very complex filamentous system that Graham Warren and his colleagues very effectively characterized since. But the concept here that's important is that each of these vesicles is attached by what we call the string. In fact, the title of our paper was Vesicles on Strings. Okay? And the idea was that a Golgi vesicle is retained locally so that when it fuses, it has to fuse up or down a cisterna, or possibly to the same cisterna, and therefore retains a kinetic memory of where it comes from. And this would provide the ability to carry out a biased random walk. Now today, we know an awful lot about these strings. They've been characterized by many laboratories, morphologically and biochemically, throughout the Golgi stack. We know where they're located. We know much about their domains. We know how vesicles bind to them. We know, in some cases, exactly how they bind Rab proteins, and Rab proteins in the vesicles bind them. There's surely more to learn, but the message is that the basic idea that vesicles are tethered by strings and that they tend to bud, when they bud from the Golgi, they tend to stay on the Golgi, is a known fact. And that is a requirement, mathematically or in any other way, for a distillation process to work. So again, what don't we know? And for the very reasons I mentioned of not being able to track vesicles, we have no idea really what the flow pattern is within the Golgi. So this is a series of potential flow patterns, the first 4 of them actually, that I lifted from a chemical engineering textbook in 1980, because it became quickly apparent that chemical engineers who deal with factories that separate materials from each other by differential fractionation in repetitive steps under continuous flow have come up with strategies that achieve this. This is called countercurrent flow. It was actually devised by Lyman and Craig for chromatography in the 1940s. It was the first chromatographic method used to separate proteins. You have a series of tubes or layers. You have a transfer process that transfers material from 1 to 2 to 3. And is selective for one component. You have a retrograde transport that transports in the opposite direction that is selective for the opposite, for the other component. And the result is component A accumulates at this face, component B, for which the dashed arrows are selective, accumulates at the other face. Its effectiveness increases exponentially with the number of stages, exactly as distillation would require, because it is distillation. A similar process called cross-current is mathematically the equivalent. In this case, the retrograde flow for component B is direct from cisterna to ER, cisterna to ER, or cisterna to ER. They're mathematically equivalent. These would be vesicles going from the rims of the Golgi back to the ER. This would be layer to layer on the whole going to the ER. And any mix would, of course, achieve the same result. You can achieve the same result if you have selection that's perfect and bidirectional. You can also have random walk and no selectivity on the part, let's say, of the antegrade cargo, the A transfers. This would just be proteins type A going back and forth in an organized way, but with a rule, always 1 to 2, 2 to 3, or 3 to 2. It can go backwards, but it can't be random. So you can imagine tubules connecting the cisternae of the Golgi rather than vesicles in a more modern version of this model. Your antegrade cargo is subject to going back and forth, but it always has to go up or down 1 according to the tubular anatomy. Or it could be a vesicle that can go both ways, restrained by a tether. We have no idea, frankly, at this point, for all the reasons I've mentioned, but this provides a very simple conceptual framework for evaluating models in terms of their efficacy. And in a way, it says, at a certain level, and this is a message I wanted to communicate, the basic problem of the Golgi is solved, which is to say, the purpose of having a Golgi apparatus. Why do cells always have a Golgi? A question I raised for you that I've been asking since 1980. It is needed for iterative sorting to continuously purify the proteins of the cell to separate the constituents of the endoplasmic reticulum adequately from the rest of the cell. Therefore, the rest of the cell is retained together until the endoplasmic reticulum has been rigorously removed in a distillation process, allowing the cell now to safely separate the residuum to the subsequent compartments. That's why cells have a Golgi. Why does the Golgi have a stack? Because the stacked morphology allows biased transfers to neighbors. Biased transfers to neighbors are necessary by whatever mechanism is physical, by whatever biochemical or morphological mechanism is necessary for multi-stage sorting. Okay? That is the principle of the Golgi. I believe that is why cells have a Golgi, and all Golgi operate according to those principles. Now the question comes, at a molecular level, that is the physiological explanation of form and function. My belief is that that part is not going to change, and in a textbook, if it isn't a textbook in 100 years, that's exactly the way it will be. But what is the flow pattern and the mechanism of transfers, that remains still, remarkably enough, after all these years, to be determined. So what do we know about it? So I'll take you back to, very quickly, back to the model that came into favor first, which is the vesicle transfer model, the idea that vesicles, specifically COP1-coated vesicles, or codomer as we first called them, are responsible for transfers that carry the anterograde cargo across the Golgi stack, in addition to their now established role in retrograde. And this came about initially through our reconstitution of transport in a cell-free extract between Golgi stacks. I went through this in some detail on Friday, so I won't go through it again, except to say that when when extracts containing two intact Golgi stacks were mixed from two separate cells and transfer was observed, it was apparent that a diffusible intermediate must somehow be involved. And that diffusible intermediate suggested a vesicle. We were able to isolate those vesicles, as I've showed you. I showed you on Friday. They have a distinct coat. We called that the codomer. But we discovered the codomer protein that coats it, and it's now called the COP1 coat. We were able to produce purified vesicles, COP1 vesicles, from a donor Golgi fraction. This was work we did in the early 1990s. We were able to separate it from the Golgi, and we added those purified COP1 vesicles containing the cargo protein to the acceptor Golgi, and they went on to fuse. That demonstrates their role as intermediates. So it's certainly true in vitro that COP1 containing vesicles can carry vesicle cargo from layer to layer, from one Golgi to another. Is it true also in living cells? So recently, my colleague, young colleague Greg Levu, this is our paper showing something similar in 1984, but very recently, we've done some cell fusion experiments. And what we do, using modern methods, is we take two cells and fuse them together, two fibroblasts, two HeLa cells. One carries a Golgi, let's say, stained in green, because it has a Golgi marker or cargo protein with green fluorescent protein. Another is transfected to have a Golgi stack that would have, let's say, red fluorescent protein on the same or a different Golgi resident or cargo protein that passes through the Golgi. These two cells can then be fused within a matter of seconds because their surface contains a viral fusion protein that's activated by a brief exposure to pH 5. This fuses the two cells into a common cytoplasm, and the nuclei are well enough resolved, and the Golgi, as I've mentioned, stays around the nucleus and the centrosomes, which remain separate, so that over a period of more than an hour, the Golgi remain entirely separate. And we can then ask, do red and green exchange or move between these separate Golgi stacks in the living cells, which is equivalent to what I described we did in the test tube many years before. And so we did this experiment, and we want to ask, is there a hybridization, and are there some molecules that are transferred and not others? So what happens, for example, if we load this Golgi with a cargo protein, and we ask, is that cargo protein transferred to the other Golgi, an anterograde cargo protein? Well, we've actually loaded in the experiment I'll show you, we've done many versions of this, two colors into this one Golgi. They're both cargo proteins. One cargo protein is a small soluble protein, and the other is a large aggregated protein of the size of collagens, artificially aggregated. I'll show you how in a moment with other experiments. That is too big to fit in a COP1 vesicle. So we have in the same Golgi small cargo molecules that are moving through this Golgi that can fit into COP1 vesicles very easily. COP1 vesicles are 70 nanometers in diameter. Others, aggregates of hundreds of nanometers of size that can't fit in. What happens to those two cargo? And what we observe is that the small cargo, this is the amount of fluorescence due to the small cargo now found in the acceptor Golgi in the other cell. That Small cargo is delivered over time, but the large cargo, which can't get into COP1 vesicles, is not delivered at all. By the way, if we chemically disaggregate that large cargo, I won't show you all the control experiments, obviously, given the time, it now is transferred exactly at the same rate as the small cargo because it becomes small cargo. The Golgi remain intact, and most interestingly, if we look in the region that is fused between these two cells, between the two nuclei, we say, what do we see? We see actually a wave of little dots moving across. And with super resolution, we can image these little dots. And in other experiments in 2 colors, these little dots not only contain the cargo molecule, they also contain the COP1 coat protein. And at super resolution, we can actually locate them both to within 100 nanometers, okay, which is approximately the size, the diameter, the outer size of the coat of a COP1 vesicle is 105 nanometers. So to summarize, COP1 coated vesicles containing the cargo can be observed moving between the one Golgi and the other in a kinetic wave at exactly the right time. We've actually gone so far as to photo bleach those vesicles as they are passing through between the two Golgi. And when we do that, the arrival of this fluorescence no longer occurs. So therefore, these vesicles were delivering the cargo from the donor to the acceptor Golgi. So this proves that COP1 vesicles can carry anterograde cargo between 2 separate Golgi, okay? So it suggests they could be involved in transport within a single Golgi stack, but obviously doesn't prove it. But it makes the hypothesis reasonable. So we have actually then a reasonable hypothesis, which is that vesicles deliver cargo, anterograde cargo to the Golgi, that anterograde cargo is transported across the Golgi by these types of vesicles, COP1 vesicles, which may occasionally fall off, despite everything that I've said. The efficiency of that transfer between Golgi stacks is about 10%. So it may be that 90% remains within the Golgi stack and 10% falls off, and then in a single cell ultimately rejoins, but gets captured, but can fall off into outer space and find another Golgi in a distant cell in such a situation. We think that in this model, retrograde transport is mediated also by COP1 vesicles. And what motivates this, in addition to everything that I've said, are immuno-EM studies that we did in the 1990s and early 2000s that demonstrated 2 types of COP1-coated vesicles. One containing retrograde cargo but not anterograde cargo, the other containing anterograde but not retrograde cargo. So morphologically, again, a snapshot picture consistent with this, okay? The critics of this model said, and we could not disprove, how do you know that these vesicles containing the anterograde cargo are actually moving anterograde in the cell? We don't. They don't come with arrows, so we can't prove it, okay? But on the face of it, one would have to say it's good evidence in favor of the model. Now there's another model, and that model was motivated by the discovery by Alberto Luini in 1998 that collagens, large natural protein aggregates that are unable to get into small vesicles, they're just too big, he asked, could they get through the Golgi? And the answer is they do. And he could see that these collagen aggregates were present at every level of the Golgi stack, and they apparently never left the Golgi stack during this passage. That point is a little hard to prove, but as best he could examine it, that was the case. How then could that be explained? Well, he actually suggested what had been an earlier model, and it's interesting how the world works. In the era of Palade and Farquhar, it was assumed that the cisternae actually move across the stack. That's why these were called forming phase and mature phase. When the vesicles were described, that went out of favor, and this was renamed cis and trans by the same people who had originally named it forming and mature. Forming because in the cisternal maturation model, these vesicles are thought to, from the ER, the COP2 vesicles, are thought to join each other to form an entirely new cisterna. Meanwhile, at the opposite phase, the last cisterna, the mature cisterna, the transmocisterna, would disappear, entirely be consumed into vesicles, have no permanent life whatsoever, so that if you were to track in any individual cisterna, it would be seen to be moving across the stack one layer to the next. So that cisterna 1 becomes 2, A minute or two later, 2 becomes 3. It's a very appealing explanation because it also explains why you need a stack. By the way, it's perfectly equivalent with to distillate, perfectly consistent with distillation. That's why I emphasize the basic idea of distillation and that the Golgi has this morphology to allow iterative sorting is correct. The only difference between the models is whether the cisterna is itself in its entirety a transport vesicle, or whether transport vesicles form from it by accretion and move it bit by bit into the next cisterna. But it is a difference, and in this model, the vesicle, bidirectional vesicle transport, you have anterograde transport by Kaplan vesicles, and the cisternae stay absolutely still. In this model, there is no transport by anterograde vesicles. All vesicles go retrograde, and the cisternae are the transport vesicles, 1 becoming 2, 2 becoming 3. I think you can see that if we could adequately visualize the proteins in relation to the morphology in real time and in appropriate resolution, it would be easy to make a distinction between the two, but unfortunately that hasn't been possible. So this has been, we now have 2 models for which there is considerable evidence for each. And the question is one right and the other one wrong? Or is it possible that, and I think it is possible, that they are both correct and that there's one mode of transport and there's another mode of transport that perhaps occurs on a different time scale and maybe perhaps at least as much related to the process of Golgi duplication and division as to transport during interphase. But in any case, it's perfectly possible that both are correct. Now, in the last 2 or 3 years, we've used synthetic biology approaches to perturb the Golgi and to test predictions of the models, because the predictions are quite different, especially as regards gluing and separating cisternae. Regarding gluing cisternae, well, let's say, regarding gluing cisternae, if you were to, in this model of moving cisternae, if we were to apply a glue in here, Velcro, artificial adhesion between this, the various cisternae, what would you expect? You would, of course, stop cisternal maturation because you could never lose the last cisterna. The Golgi cisterna should get stacks should grow bigger and bigger, and transport of anterograde cargo should come to a halt. It's a very clear prediction. On the other side, if you were to separate the cisternae so that this cisterna no longer had any spatial relationship to the other cisternae, again, this cisterna could not become that cisterna, and cisternal progression would cease, and maturation would stop, and therefore transport would stop. In vesicle trafficking could be slowed, but in principle it could survive both of these maneuvers. Since the cisternae are already permanently adhered in this model, simply adding more adhesion shouldn't stop vesicle flow. It may make the membranes a little more resistant to bending, but it should allow vesicle transfer to continue. Likewise, since vesicle transfer is inherently distributive, although the transfers may be a little sloppier, if you were to separate one cisternae from another, you would not expect vesicle trafficking, anterograde transport to stop. An interesting third variation, oh gee, I'm running short on time, so I better make this very short. Another approach is to control the shape, is to introduce a marker. So let's go back to this model. In this model, if I could mark this cisterna, that mark would always go forward, right? In this model, if I were to mark that cisterna, that mark would never go anywhere, right? The problem is, what sort of a marker can you put in here that can't go anywhere? Well, actually, we created a marker which is called a staple. The staple actually adheres across the luminal side of this cisterna. It actually links internally the two opposed membranes together. If you have such a link, there's no way it can ever be packaged into a vesicle, because it being linked to both membranes, the entire cisterna would have to become the vesicle, and it's too big. Well, we've created exactly such by controlled adhesion. These are domains, protein domains that self-adhere. It's a mutant version of an immunophilin called FM, which we've used in many studies, as many labs have. And we put repeat domains onto membrane proteins or not. And the key point is that the FM domain has the molecular property of binding to itself in an anti-parallel orientation in a way that is prevented or easily reversed when you add a ligand. And using this system, we've developed a series of designs. These are electron micrographs. If we express in the endoplasmic reticulum simply a soluble version of an FM domain, unconstrained by the membrane, then it will self-aggregate like an immunoprecipitate to form a sphere. And those spheres are unattached to the membrane. If, on the other hand, we attach, put the FM domain in the lumen and we attach it to the membrane by a membrane anchor, then it actually will form, like a cadherin, a staple across the lumen. This is an endoplasmic reticulum membrane whose lumen is stapled. We can actually orient the same molecule towards the cytoplasm. I won't show you any details on that. And if we do, we now get our staples where we're stapling one ER lamellae, this is the lumen of one, stapled to another. So now the staple goes across the cytoplasm. So there are many different constructs that you can make with these sorts of synthetic approaches. This is an example where in the very same Golgi, I'm just showing you two different cisternae that show one a staple and the other a ball. So we can actually express these aggregates, aggregating proteins, in the very same cisternae. And we can trap them kinetically, I won't go through how, with a temperature block in the cismos cisternae, and then we can release the temperature block by going from 20 to 37 degrees. And what we observe is that the membrane staples of the Golgi cisternae always remain over time in the cismos cisternae. Very few get into later cisternae. On the other hand, even in the same cisternae, the spherical aggregates move very effectively forward to the trans cisternae. So what does that tell us? That tells us that a staple in the center of a Golgi cisterna stays put, but a Luini-like collagen particle in the rim of the Golgi moves forward. So I think we've resolved the paradox of how a collagen molecule, an aggregate, can move from one end of the Golgi without ever leaving the Golgi stack, from one end to the other, without the cisternae moving forward, because actually the rim is what moves. We think that the collagen aggregates and the like are found in the rims, where they are, remember, the centers are flat, and the adhesion of the Golgi forces them to be flat, and because they're flat, they squeeze out anything big and round. And that is why these aggregates are, and indeed have to be observed physically, squeezed out to the rims. So if the rims are somehow able to progress 1 to 2 to 3 by a process of fission and fusion, if that were to occur, then the staples in the center and the Golgi cisternae as a whole can remain stable, and our observations in Luini's can be very easily rationalized, perfectly consistent also with much smaller vesicles which can't carry the large cargo, forming between Golgi cisternae, and indeed in the cell fusion experiment carrying the small cargo. And in fact, we observed quite some time ago exactly these structures. We call them megavesicles. And when a wave of large aggregate is moving across the Golgi, these actually can be seen to transiently carry up to 40 or 50% of the cargo at that time. So we called them megavesicles rather than moving rims at the time, but that's exactly what we think that they are. Okay, what about gluing cisternae together? And I'll try to be faster here. We've used an analogous set of dimerization proteins. The FM domain automatically binds to itself. The drug breaks it up. FKBP and FRB, also invented by Schreiber and Crabtree, are two protein domains from larger proteins that do not naturally bind each other unless a dimerizing agent is added that acts as a joint ligand, a small molecule ligand between the two of them. And when it is added, a molecular dimer is formed. Fortunately, the drug penetrates cells, it's easily removed, and so you can cause proteins to dimerize and undimerize upon command. What Greg Levue and Julia Dancourt did, it's not yet published, we're just writing this up, is they joined these proteins to the adhesion proteins in the Golgi. So we took proteins that we know from Warren and other studies to mediate the adhesion of the cisternae of the Golgi to each other. And without bothering them with molecular names, there's one that's in the medial cisterna, there's one that's in the cystisterna, and one that's in the trans. And what we did was we made a peanut butter and jelly sandwich in which we introduced artificial glue. So we took, for example, the FRB protein here, and we put it on the trans-cisterna attached to a natural adhesion protein, and also in the cis. And in the middle, we put its protein partner, FKBP, attached to a protein in the middle of the Golgi stack. So now nothing should happen until we add the dimerizer, in which case, depending on the concentration of the dimerizer, we can ratchet in more and more adhesion, putting a glue that should span the Golgi stack. Does that make sense? That's what we did. So this is now excessively gluing the Golgi stack. When we do that, we do in fact excessively glue the Golgi stack. This is immuno-EM against a common constituent, a tag on all three of the glue proteins. And you can see that the glue is exactly where it ought to be at the interfaces of massively adhered Golgi cisternae. And these Golgi are perturbed from their flat state, probably due to the excess of adhesion. We also can, by the way, isolate these complexes that are predicted. These complexes that are predicted by immunoprecipitation from the extract. We've done photobleaching, and whereas these constructs are mobile before the dimerizer, they are completely immobile after. So we are absolutely confident that we're forming the correct complexes that we say that we are, and we're confident of their localization in the cell. And now, when we measure transport, and these are now individual experiments. So each is an individual Golgi area. So we're looking at a single Golgi stack in a single cell where we have overexpressed these proteins and where they are hyper-adhered. And we're looking at a wave of cargo, which is released with another trick that I won't go into, from the Golgi into the ER. And this is the content of fluorescent cargo versus time after release of the cargo from the ER in a synchronous wave. Here are control cells, and you see the cargo goes into the Golgi and then it goes out. And here on the cell surface, in the very same cell, you see it appears. And you see how actually reproducible the data are. This is the standard deviation. These are the means. And each is an individual Golgi in a different cell, averaged among 4 different experiments. So this is highly reproducible. And you can see the wave goes into the Golgi, out of the Golgi, onto the plasma membrane. These are the glued Golgi. It actually, it takes about twice as much time for it to come out of the Golgi, and most of that delay is actually to the surface, and most of the delay is going into the Golgi. So evidently, even though these Golgis are extensively glued, transport of anterograde cargo can still occur, though at about half the rate. And of course and I should mention, by the way, that aggregated cargo's transport is completely blocked, and the collagen-like aggregates, including collagen itself, in these cells are stuck 100% at the first cisterna. So even rim progression is blocked. So this suggests that vesicle transfer can occur in Golgi that are made permanently attached. Rim progression, when you attach the cisternae perhaps here, cannot occur, but anterograde transport can occur at perhaps half the rate. Separating cisternae from each other in a rigid matrix. I'll be even quicker here. What if you do the opposite? You add an adhesion process to another organelle that never normally occurs. So we'll take our proteins that will stick together with the dimerizer, we'll put one on the mitochondria and the other on an adhesion protein in the Golgi. Okay? Now what should happen? If you add the dimerizer, the mitochondria should stick to the Golgi, right? And as you add more and more of the dimerizer, what should happen is the adhesion energy to the cisterna will eventually exceed the adhesion energy from cisterna to cisterna, whatever that may be. And eventually, a cisterna should be encased within a rigid framework of mitochondria. And these dissected cisternae we refer to as land-locked Golgi cisternae because they do not move. They actually stay in place for the better period of an hour, even at super resolution. Are they capable of transport? So here's what they look like before the dimerizer, after the dimerizer. They're not all perfectly one cisterna, but by no definition is the Golgi morphology or any of the previous relationships left. Remarkably enough, those landlocked Golgi and the intact Golgi receive the wave of cargo at exactly the same rate, and they in fact they both transport out of the Golgi. Here's the control. Here's the landlocked Golgi. The landlocked Golgi are as efficient at transmitting anterograde cargo towards the plasma membrane as the normal Golgi. It's just that they take about twice as long. To me, this is just absolutely amazing. And how does this happen? We're obviously studying it right now because landlocked Golgi give us our best chance today to track transfer, because we have separate Golgi cisternae, which are easily resolved in the light microscope, and maybe we can track vesicles or tubules going between them. And you see, for example, at the light level, this is optical imaging where the green are individual COP1 vesicles, and these are the purple here are individual Golgi cisternae that are encased in mitochondria. And you can see the vesicles have separated actually from the Golgi cisterna, the COP1, and can be seen at various places in between them. If you look at an EM section, these are actually serial sections. You'll see in serial sections vesicles of the appropriate size, 70 nanometer size, and you'll see that many of them are coated buds. Sometimes they're seen separate, sometimes they are budding, but in any case, there's good morphological evidence, and so we think, but we don't know for a fact, that the transfer between these landlocked cisternae is mediated by COP1 vesicles, but we shall see over time. So summarizing then, whether we actually whether we do away with adhesion of cisternae to each other, or we prevent their dissociation from each other, either way, basically transport can occur. And so anterograde transport, and that's hard to reconcile with a model which is principally dependent on the movement of Golgi cisternae from one end to the next. Now, does that mean that that sort of movement does not occur? No. It could very well contribute to anterograde transport, and in some organisms it may even be quite important, perhaps even the principal mechanism. So I don't want to overstate the case, but I do think that this establishes, at least in my mind without a doubt, that cisternal maturation or progression is not a prerequisite for anterograde trafficking through the Golgi stack. And the reason I emphasize that conclusion is that if you pick up any textbook today, it will describe anterograde transport as a fact being as being mediated by cisternal maturation. And it certainly that overstates the case. So how then are we ever going to resolve this dilemma? And there's two possibilities. One is that we never will, because what will happen is that people will get interested in other problems, and no one but old people like me will be interested in resolving this problem anymore until some amazing new method comes along. That's actually what happened, if you remember, with protein synthesis. By 1980, nobody was working really as a frontier on the mechanism of how the ribosome works. The whole field stopped, okay, until we got crystal structures of the ribosome, and then it started again, okay, and we learned ever so much, and it's so beautiful. Super resolution microscopy, I'm hopeful, will become our version of the crystal of the structure of the ribosome. And at Yale, we have a nanoscopy center that is currently supported by the Wellcome Trust, and is really, our goal is really ambitious. Our goal is to achieve three-dimensional resolution at 20 nanometers with two colors in real time, meaning 100 millisecond dynamic frame rates in 3D. And what we've succeeded in so far is 2D, two-color, real time, both by STED and by Palm, but not yet, not yet 3D dynamic. We have 3D two-color with X, Y, and Z, all 30 nanometer or better, but not real time. So what we now need to do over the next several years is figure out how to do it 3D. What you're looking at is, and this is done with a combination of things. We build our microscopes, we have new optical approaches, but mostly what we're doing is doing what a lot of sort of physically oriented, leading laboratories, we don't claim to be inventing microscopes. We're not really doing that. Maybe somewhat on the edge, but mostly what we're doing are system integration. And we're really making these systems work for cell biology by getting combinations of dyes and tags, okay, and adjusting the imaging and the lasers to what the biology requires, and adding in algorithms and analytics that are really worth a lot. So this is an example. In 2D, the frame size is 2 seconds, largely the work of Jörg Beversdorf and his colleagues, with whom we closely collaborate. This happens to have been done by two of our young colleagues. These are actually in real time mitochondria and endoplasmic reticulum elements. You'll see the space of the cristae in living cells. Okay, the resolution here is about 30 nanometers, I believe. You can just barely make out, but if we were to take static images, you would easily make out the lumen of the endoplasmic reticulum. This is an example from our laboratory, together with Jörg Beversdorf, of single COP1 vesicles budding at super resolution. This is a single Golgi cisterna, a cis cisterna. You can see where it is fenestrated, okay, even. I hope you can see the darker areas. That in the electron microscope would be called a fenestration. This is a tubule that forms, the tip of which has a COP1 coat. This cotomer has been labeled at the normal level of expression by CRISPR incorporation, and it's an indication of what's going to be able to come. So I would expect within the next several years we will be tracking COP1 vesicles at super resolution. This is in 2D. It is again at 20 nanometer resolution and at 1 frame every 2 seconds. So what will be needed to take this sort of effort from its current infancy to really become contributory to not just this area of cell biology? As I've mentioned, it's going to require going from 2 to 3 dimensions. And let me put a number on that. You might say, well, you can do 3 dimensions today in a static image. Why can't you do it in a dynamic image? Well, of course you can, okay? But you can't collect enough photons to get enough resolution. So if you could collect all the photons you wanted, and if you by the time you come back and iterate your scan, too much time has gone by. Or you could use a lot more light, but then you will have photo-bleached your sample. So that's the basic problem in the field. That problem is to a large degree unsolvable. Yes, we can improve the brightness of the dyes or some of their properties, and from our experience, that's something that will give a factor of 2 here, 50% there, sometimes a factor of 3, and if you get enough of that, you might even get a factor of 10 with new chemistry over a number of years. My colleagues who are experts in this estimate we need a factor of 50 to 100 more photons to get dynamic imaging in the time frame that I've just mentioned without photo bleaching, which is to say we basically need to be able to get good images that are on the order of 100 times better with the same amount of light that we presently have. And how are we going to do that? That's the problem. Okay? So the approaches that we're starting to take now and it's basically taking less light and using it better. Now, and the way I look at it, and it's not a very sophisticated way to look at it, is that maybe what we need to do is assume we know some biology every time we look at something. So in other words, here you're looking at this image, it's as though you've never seen a Golgi before. You're taking into account nothing that you know about the Golgi, what molecules it contains, where they're located, or what its shapes are. You're assuming none of the above. Okay? But actually, we know what COP1 vesicles look like. We know what Golgi cisternae look like. Yes, there are many possible shapes, but there's a limited number of ranges. We also are tagging molecules. We know that they're in a membrane, not in a membrane. They're in the same membrane or a different membrane. And though they may be scattered according to an unprejudiced view, we know actually they're in a plane because they're on the same membrane. So that's the type of biological knowledge. For example, let's suppose we were taking a picture of a soup can, a can of soup with an iPhone. If we were doing that, you might want a perfect picture, but if you already knew what a soup can looked like, you might just get a slight shadowy edge of it with a few photons, and then know from previous information, biological information, what a soup can looks like and fill in the boundaries. So that's the type of thing that we're trying to work on how to do, and it is not easy to develop those forms of image processing. And finally, the last thing, I realize I'm seriously over time, is you have to show membrane outlines. A serious deficiency in super resolution is you get a bunch of dots. Okay? If the dots are oftentimes not so close to each other that you can really say it's a membrane outline, more often than not. So if you're looking at cargo molecules, how do you know what it's on? For example, if you were looking at a Pizarro painting, and there's 3 colors, and if it's a scene where you're looking at, let's say, a boat on the water in the shore, that's a typical Pizarro scene. And if in your mind's eye, you didn't know what you were looking at or you couldn't relate it to previous images, you just see a bunch of colored dots. Imagine seeing that in 3D and they're moving in real time. Okay? That is what you have to face up to if you're successful. How do you solve that problem? You need to come up with an optical way to actually see membrane outlines as if you were looking at an electron microscope. Well, we don't have that yet, but Derek Toumre in his laboratory at Yale are developing it. by using lipid-based dyes that, for example, are based on ceramide, where you can incorporate the ceramide into the ER or the Golgi in living cells at high density, so that about 1 to 5% of the lipid molecules are labeled. So it's very high density. We can do this with either STED or with palm blinking probes, single molecule imaging probes. And then once we've got those incorporated, then a dye can be clicked in with appropriate properties, and even in the living cell we can image membrane outlines. And we've started to make progress. This is actually this is real time. You're looking actually in the living cell at the entire endoplasmic reticulum, outlined in its membrane outlines and contours, seen at lower resolution on the left and higher resolution on the right. And I hope even here you can begin to see the hollows in the tubes of endoplasmic reticulum. So for me anyway, this is fairly breathtaking. You're looking at the dynamics of an entire organelle at super resolution. This is at in this case, at 40 nanometers resolution. We think we can do better, but this is what we have today. And the speed is adequate to measuring most membrane transfer events. So this is what's coming. And in the next 5 years, I would be surprised if our group or other groups do not succeed in dynamic three-dimensional imaging with this or perhaps other techniques. And I just want to finally close by paying appropriate respects to the people who contributed. Greg LeVue is the leader of the synthetic biology approaches, all of them that I presented to you. Minhua Dunlap is a senior postdoctoral fellow who has developed further developed the landlocked Golgi and is working on that currently. Patrina Pellet and Lena Schroeder contributed importantly to the cell fusion studies involving transfer between Golgi apparatus, which were also pioneered by Greg LeVue. The older work was the result of a very successful, but long ago, no longer fresh collaboration with Lilia Orci and our colleague, young colleague then, Alan Volchuk. And finally, all of the super resolution work is done in collaboration with the Yale Nanoscopy Center, which I've described to you, which is at the moment primarily work with Jörg Beversdorf's laboratory and Derek Toomray's laboratory, and increasingly a young recruit, David Baddeley, who's going to be is starting to do the algorithms and image processing. And I have to say, it's amazingly, just before leaving for the trip, I saw a trick that this brilliant young guy pulled out of his hat that was good for a factor of 20. So I'm really optimistic. And that's just processing the information. So I hope sometime, if you were willing to have me back to talk about this, I'll be able to, in 5 years, show you pictures of vesicles moving around the cell that do have arrows and where we can measure their cargo, and we'll finally have some answers. So thank you very much.